Today we're going to be talking about the Watson and Crick DNA structure. A previous study found that for every thymine there's an adenine, for every cytosine there's a guanine. This is called Chargas' rule. So Watson and Crick came up with the idea of base pairing, where thymine pairs to adenine and cytosine pairs to guanine. They're paired by hydrogen bonds. Hydrogen bonds are pretty weak. They're not really the main force keeping DNA together. They're more important for specificity. C and G both have 3 hydrogen bonds, so they know to bond with each other because there's 3, and T and A both have 2. This is how the base pairs know how to bond to each other. Next is the composition of ribose sugars. The difference between deoxyribose and ribose is deoxyribose is missing an oxygen on the 2' carbon, hence why it's called deoxyribose. Ribose does have an oxygen on the 2' carbon. Almost all of the carbons in deoxyribose and ribose play an important part for nucleotide bonds. The first prime carbon attaches to the nitrogen base. The second prime carbon, as I just stated, distinguishes DNA from RNA. The 5' carbon has a phosphate, and the 3' carbon bonds to the next 5' phosphate. This is how the sugar phosphate backbone is made. In the sugar phosphate backbone, the 3' carbon of the first nucleotide bonds to the 5' carbon of the next by a phosphodiester bond. These strands run anti-parallel of each other, and they run from 5' to 3', as you can see. Here's a 5' carbon, here's a 3', 5', 3', and then in the opposite direction, 5', 3', 5', 3'. We're always adding to the 3' carbon. They also determine some other forces that help stabilize the double helix. One is base stacking. This is when base pairs lie perpendicular to the sugar phosphate backbone. Adjacent bases have an attraction that helps stabilize the double helix, as put right here. These are not base pairs, they're next to each other on the same strand. And there's an attraction between them that's helping stabilize, but it's still not the main force stabilizing the double helix. What's really stabilizing the double helix is hydrophobic interactions. The outside of the double helix is the sugar phosphate backbone. This is hydrophilic. It likes water. The inside of the double helix is the nitrogen bases. This is hydrophobic. It does not like water. So the hydrophilic outside shields the hydrophobic inside by coiling planktonically. There's also some other key details of the double helix. We have our smaller minor grooves and our larger major grooves. There's also handedness. The normal form of DNA is exclusively right-handed. As you can see here, the DNA is moving upwardsly to the right. Even though I stated that DNA is exclusively right-handed, that doesn't stop media outlets from doing left-handed DNA. So you can see this goes upwards to the left, up to the left, again to the left, to the left, and to the left. All of these are incorrect forms of DNA. Some key points to remember. A always goes with T, and C always goes with G. Ribose carbons all play specific roles in the bonding of nucleotides. Hydrophobic interactions are the main force stabilizing the double helix. Hydrogen bonds really just provide specificity to the double helix. DNA strands always go 5' to 3', and phosphodiester bonds are what really keep the sugar phosphate backbone together. Thanks for watching!